Okay, my friends, this is, again, as serious as it gets. This is about autism. This is one of the most disastrous things that's happened to our society. 1 in 40 kids in the United States has autism now, and they're trying to focus in on what causes it. They really don't know. So they say they uncovered a key factor in the brain development. Now, they're going from the moment the brain starts to develop. And I always start at the beginning too. And it's saying that one of the most studied genetic causes of intellectual disability is a mutation in a gene. All right, remember this, a mutation in a gene that was originally called LIS1. Well, what is a gene and what is a mutation? Okay, here's how a doctor thinks. If a gene isn't working, he says, oh, that's a mutation, your parents didn't pass that on to you, and, it may run in the family. And that's true, because you can have a changed gene, and it will carry on. Yes, absolutely. What you want to do is be sure that your genes are good. Now, how do you inherit good quality genes? Your parents have to be healthy to begin with, and they have to have a pretty good genetic sequence. Secondly, when you are literally at the moment of birth, when you touch the environment, you become programmed. All of this stuff starts getting programmed. And it happens from the vaginal juices. First of all, you're in a sealed environment. You're all cleaned up and nice and nothing happens to you. And you're getting your nutrition from your mother in the womb and all that business. And all of a sudden, you're ready to go. Out you come through a bunch of bacteria. And that's exactly what it's supposed to be. It's supposed to be bacteria. And I got to be honest with you. If I was a woman and I was going to have a baby, I wouldn't take a bath for a couple days before having a baby. That's just research saying that there's going to be a lot of extra bacteria in there, and that's what you really want. Even the feces, when you have a baby, they're right next to each other, and they always intermingle. Well, not always, but they should. Now, having a C-section is you need to swab them with this kind of stuff. I was thinking they should make some kind of preparation birth baby juice, something that you could just slaughter all over them, and they'll have all of this. They'll see these codes, because all it is the genes are set to go, but they're not going to. They say, well, there's nothing out there for us to do. And they say, whoa, didn't you see that bacteria? No, I didn't see it. Oh, what are you, a C-section? Yeah, I'm a C-section. Oh, obviously you didn't see it because it comes out of the vaginal canal or the feces or whatever. Now, I think they're doing swabs of the babies, that are C-section, I hope, because that's where you're getting your program codes from. So now we're off the case of genes. We know genes make either assembly codes, which is just like a program manual, how to make your fingers work and all of that stuff. I mean, it's very, very, very complicated. Or they make products, which are enzymes and catalysts. Well, what does an enzyme and a catalyst do? Those are the things that create chemistry. If you don't have them, you can't create the chemistry. What does the chemistry do? The chemistry breaks down food, so you can digest it and you can get the nutrients that you need. Or the chemistry goes out and attacks invaders, because they are going to come in with a bag of tools that you better have a better bag. And if your genes know what to do and they recognize that invader, you're good to go, because you got a bunch of guys. You got a ton of guys. You're not going to get invaded by a bazillion, trillion guys all in one shot. If you get deep wounds, yeah, you got some problems. But normal scratches and cuts and all that business, you guys can go there and pop those guys off in 10 seconds, case closed. But you got to have your genes working. They have to have their enzymes. They have to have their chemistry. They have to have that gene turned on to say, if I see a certain sequence of proteins come in, I know that's a bad guy. And I know it's a bad guy. And I know how to get rid of him. I know how to kill him. And here's the sequence to do it. When I see that happen, this automatically says, go make this stuff, squirt it on those guys, and you're healthy. Okay, my friends, this is very, very simple, but not understood. Genes are segments of the DNA, double helix, and there's a ton of it. It's all balled up in the coils of these little tiny codes, and they're program codes. That's all they are. Now, when that gene is turned on or turned off, or functional or non-functional, it creates two possibilities. One is it can create a product, which is nothing more than a chemistry set, which is called a protein. I will show you those in a second. The other thing it does is it can create instructional codes for how many fingers you have and how your eyes work and all that stuff, because all of that is built into this codes. Your eyes are different than a snake and so forth. That you see different ranges, you do all kind of different things. Your lenses develop differently. Your toes, your fingers, your claws, whatever you may have, is a product of this gene. If it's turned off, you don't get whatever was supposed to be there. It's as simple as that. And basically they are on-off switches, genes. Think of it that way. And they create products, which are different chemistries, or they have instructional, like a production manual, how to assemble your foot and how to make your eyeballs and all that stuff. Because those, again, I think I went over that, but I like to, really make you understand. It's basically programming, and there is so much program code in here. It's like, like your smartphone now. Oh yeah, you've heard gene editing. That's all they're doing is going in and changing a couple of these codes so that it turns on or turns off. It does something that it shouldn't do, and that's how they do this genetically modified foods. They come in and they say, "Oh, don't worry about being poisoned. Just keep growing, no big deal." All right, before I start rambling on about genes and so forth, what you have to understand is DNA is where these gene sets exist. Now, in that DNA, which they only say about 2% of it, is being used to do these things, which is to make instructions to make molecules called proteins. But other ones do not code for proteins. Those are the ones that are instructions how to build your body. The ones that code for proteins are the ones that make proteins are enzymes and catalysts and chemistry sets. And if you don't have those, you can't break down products in your body. You just can't do it. It's not going to happen. So you can't get the nutrition you need, or you can't get the defensive system going on in your interstitium, which is the layer of membrane now. And everybody's talking about it now because it's been for the last 4 or 5 years, it's been a really hot topic. This specific layer is a fluid-filled highway. I'll show it to you in a second. And that is where all of these proteins live. And you have to have bacteria to make the proteins. This is the key. The DNA can be perfect, and you're still not going to work because you don't have the bacteria to code for the proteins. The DNA is just an instruction set. That's all it is. It's a program. So it programs to say, hey, go get the Lactobacillus, head him down there, grab some niacin, a couple of sulfurs and so forth, and go up and drop it on that guy. And he said, whoa, 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 whoa. Lactobacillus, he's off the job. He said, what happened to Lactobacillus? Well, Lactobacillus, the guy took some antibiotics to kill some Lyme disease, let's say. And it killed Lactobacillus at the same time. We just can't get him back. I don't know. He's just not coming back to work. So no Lactobacillus. I can't give you the enzyme you want. I'm sorry. Well, what are we going to do? Well, I guess you're going to be pretty sick. And that is what happens. The gene's okay. The gene says, I'm working perfect. Just go get the Lactobacillus guy. See, he's done. These cases closed for him. Well, can't you eat a pill? Yes, you can, actually. You can. Called probiotics. And they don't necessarily repopulate your system, your interstitium, with new bacteria, although that is ideal. However, at least if you can supplement the enzyme, which is the particular chemistry set. If you can put that back in using a pill, and you have to take them daily, but all they are is the same thing your body would make anyway. It's just that you don't have the gene that's to say to make it, or you don't have the bacteria in you that's living in you that is the production factory. Because that bacteria are the production factory of enzymes. All right, you got to put the pieces together. Bacteria are actually the factories that produce enzymes. If you don't have the correct enzymes, I don't care if your gene is working or not, you can't make it. The bacteria is not there, it can't make the enzyme. The enzyme is the thing that does the job. So bacteria is the key to this whole thing. All right, I might as well just throw this in. There's other places in these things that are called Cpg Islands. And all they are is little sections and codes that are not programmed. They say they're uncoded, they're junk DNA, they don't know what they do. A lot of these Cpg Islands. Really what they are is USB sticks. And every one of those islands can become what they call methylated, which means it's turned on. If it's not turned on, it doesn't do anything. If it's turned on, it will turn on a sequence of events based on what that encounter was. So that chunk of worthless DNA now becomes a memory. All right, it may maybe become a program, because let's say when you see COVID, when you become immune, well, how do you think you become immune? You think all of a sudden it just stops? No. There's a gene here. One of these genes will have said, oh, I know what happens when the COVID comes in. I've been instructed by bacteria. Bacteria do the job. The bacteria went here and it says, okay, I know what's going to happen. First of all, I'm going to see 2 phosphates and a this and a cytosine and a la la la, and then I'm going to see 3 prime 5s and so forth, and then I'm going to see some tellurase, and then when I see that code, I'm going to know that's COVID-19. And what I'm going to do is this right here. I'm going to turn on the some kind of enzyme and some kind of catalyst, and I'm going to grab a hold of a couple of these type of bacteria, and I'm going to get everything made, and then we're going to send it out to kill those guys. That's the way you become immune, and that locks in and stays locked in, and it doesn't go away. So if your immunity's coming and going, you're losing the program. Or the program is not locked in. The program is existing outside your genes. And I believe that's what's happening when they give you these vaccines that are supposedly these RNA vaccines and so forth. I'm not sure they're binding to the actual chromosomes and the genes. They're just floating around in your system, and then they wear off. I think that's what it seems to be, because you're not getting the immunity like some immunities you get for life once you get it one shot. This is what it looks like. I'm just sort of speculating. Now, this is research. I'm a researcher, not a doctor. But I've gotten vaccinated, yes. However, I have pretty good bacteria in my body because I take probiotics every day, usually twice a day. And I take prebiotics, which I take Metamucil. I'll just tell you a fact. And the probiotic I use, I like Primal Defense, seems to work well. I take other ones too, so I'm not advocating anything special. And I'm sure they have designer ones that are very good. And things are not cheap. These are the things they should give to people, to everyone. They should make them free to everyone, because this is where health starts. And it's just starting now to be understood how important the bacteria is. Without that, you're done. Okay, my friends, this is going to be fun. Everybody knows Caesar. All right, he's my buddy. He's a goose. And he petrified in the Mudfossil manner. Even his feather pattern and everything is still visible. And all of the outside membranes, coatings, skin, everything that is outside of you on the edges is made of collagens, and collagens turn into feldspar. The inside is not feldspar, and his artery that runs up to his head to feed it with blood was filled still with blood, because it broke off. I don't know, I must have broken it off when I found it, or I don't know, it was laying like this, whatever, I can't remember, a long time ago. But the neck is still in here, and if you know how to look at the patterns and so forth, you can see that it's in there. And when you look in the microscope, now we're going to look at actually, this is the best thing that could happen. Catalase is in the blood, and it's a reactive enzyme. It creates oxygen like just unbelievable amounts, but it's only found, it's a common enzyme, it's found in nearly all living organisms. Living organisms. The catalase, decomposition of hydrogen peroxide to water and oxygen. Very important enzyme. Bacteria is the factory of the enzymes. So, if this is filled with blood, this artery, could the enzyme still be active? Well, if we put hydrogen peroxide on there, will it exude oxygen and turn it into water? Guess what? Yes, it will. And I can show you that in a microscope right now. So, not only can we prove my mud fossils are real, and Augustus Caesar here is a real goose, and was living at one time, and still has catalase in it and blood, that we should be able to demonstrate if what I'm showing is true. And at the same time, we can understand if you don't have the right bacteria, you're not going to get catalase. All right, if you have weak bacteria, because the bacteria create the enzyme. The enzymes are only created from bacteria. Bacteria is the factories. So whatever the enzyme you need, and there's thousands of them, because they are nothing more than a very, very, very exotic chemistry set. I mean, look at this. This is catalase. And it squirts out all in this big long string of codes. And if it's not just exactly like that, it doesn't do the job. And it's magnetically attracted. They call it magnetotactic bacteria now. Well, anyway, it gets a little deep, but trust me, this is not just squirted out from anywhere. So if Caesar, my good friend here, starts spewing out oxygen, we are going to know that it was loaded with catalase. And if it was loaded with catalase, he was alive, or she. All right, this is the microscope. That is the artery. And we're going to be looking at it up here. I have to turn the lights down right now. It's going to be kind of hard. Well, I left it up like this so you can see something. You see all these scratches in here? Whenever I look at the blood and so forth, I got a couple different types of microscopes. I scratch it all out and take it out of there. So what you're looking at there, that red spot, I believe is going to be where the artery area is. It appears that they feed up like a tube, and I believe this is the vein side coming back. But we should see blood everywhere here, because we should see catalase reactions. But primarily you should see it from where the source of the blood is, which is the artery or the vein. So I have to turn the lights down. Let's get ready. All right, listen to this. This is a little bit technical, but not much. Catalase, as you will see in a second, is a common enzyme found in nearly all living organisms exposed to oxygen. It's found in bacteria, plants, animals, the whole 9 yards. Living things. It catalyzes the decomposition of hydrogen peroxide into water and oxygen and releases an oxygen bubble. It's a very important enzyme in protecting the cell from oxidative damage by reactive oxygen species, ROS. All right, catalase has one of the highest turnover numbers, wait till you hear this, of all enzymes. Enzymes make things go faster. That's all they do. They do normal chemistry, but they make it like on rocket ship speed. One single catalase molecule can convert millions of hydrogen peroxide molecules to water and oxygen each second. And you're going to see this actually happen. Wait till you see it. It just flipped my mind out. I couldn't believe it. Somebody told me to try it on my mud fossils, and I did, and it just, as you're going to see, using my good friend here, Caesar. All right, Caesar, you don't mind, do you, buddy? Nope. Okay, thanks. Now, What do we got? Catalase is a tetramer. 4 polypeptide chains. Each of them have more than 500 amino acids in a chain. It contains 4 iron-containing heme groups. And that's where you get your magnetobacteria, I believe. Allow the enzyme to react with hydrogen peroxide. The optimum pH for human catalase is approximately 7. And then it goes on to talk about different things. Now, let's see it actually happen. And we're going to see it happen. And my friend there, Augustus Caesar, we're going to be looking right there at his artery. And here it is right there in the scope. And now we got to turn the lights down low. Okay, I used this stuff on here the other day, and it was just unbelievable. It should still have quite a bit left in it. But I believe that's blood and blood, but it's going to be blood everywhere because that's what feeds the tissue up there. Now, you see this stuff on the inside? This is the inside of his neck. It's nothing like the outside. The outside is feldspar. The inside they would call basalt or something else. Now, let's put a little hydrogen peroxide on here and watch what happens. All right, here we go. Get ready. There goes the hydrogen peroxide. Let's just keep your eye on it now. Watch. Look at you see that white stuff starting to show up? You see the white stuff starting to show up? That's all hydrogen peroxide. You see it bubbling? I'm going to come right down on top of it. Hold on. All right, this is really very interesting. This is the fluid-filled highway. That's the interstitium. Look at it move in that layer. I didn't realize that was happening. Look at it go. That's the layer that has all of the reactive oxygens. Now, there's a bunch of it coming out of here and all these other places too. But that is the layer. Now I'm seeing something I missed the other day. Let me see if I can focus in on that. That's incredible. That is the interstitium layer. That's amazing. I find something new every single time. It never ever fails. Okay, within like 30 seconds, all of the oxygen was gone. Now let's put some more hydrogen peroxide in there. This converts something that you just saw is highly reactive, millions of times a second. Let's put some more in and see if we can get more reaction. All right, look at it come out. Look at that. Look at the reactive oxygen being bubbled off of that interstitium layer. That's the key now. Now I got something to look at. But you see that? That's before I told you where the red spot was. All right, it's converting all that over and it'll turn it right into oxygen. Let's just watch it complete its process. You look at it bubbling out of the arteries, really, and veins probably, wherever they are. These feed these interstitium layers. And these are the membrane layers. You see that? That's exactly what they are. They're membrane layers. That is amazing. And they call it the fluid-filled highway now, the interstitium. Look at that stuff bubble. Because these little tiny things, if we could get really way down close, and I don't know if I can or not, but this is going to take a second to do, so let me get as close as I can possibly get. Look at that. Isn't that fascinating? It is to me. Look at that. All of that is membrane. Around, you see this? This is all around the vascular network. And that is the interstitium layer. And they said it was a fluid-filled highway, and it's apparently fluid-filled with catalase. Well, there's a lot of catalase in there because we see it reacting. Now, you see how it's almost starting to finish up reacting with the hydrogen peroxide. The catalase is that reaction is now really coming close to an end. And before long, it'll just be gone. And it'll look like somebody just had put water on there. That is just incredible. Now, if I put more more hydrogen peroxide on there, it would come back and start reacting again. This stuff is so incredibly reactive. Look, now it just looks like water. You wouldn't know it was bubbling off anymore. You see it? Basically done now. Now, let me focus in just so we can get a little closer if we can. That's as close as we're going to get. Now, let me see if I can. There we go. There we go. Now, don't forget, where are our membranes? They're around the edges. Where's our interstitium? It's up here. I can't really get to that too much. But we can see that's the interstitium layer. Now, we're going to put some more hydrogen peroxide on. Look at, right now, you have no idea that was nothing other than water. Now, here comes the hydrogen peroxide. So it will find some more catalase to do this. Look at it, boom. It's just like a rocket ship. It's all over this stuff. Look at it. Isn't that something? I am just shocked. I got to back this off. There we go. Look at that. Now the flashy stuff here, that's the lights shining down on it, reflecting off the surface. But this is from the interstitium. Absolutely phenomenal. And that, again, you see it? Right there, that's the membrane. So these are membrane areas. That's another day in paradise. Absolutely incredible. And you can see how much reactivity. And they're right about how fast it works. Here's what it says about it right there. You can read it yourself. Convert millions of hydrogen peroxide molecules to water. A single one of those catalase ones. So I don't know how much is there, but it seems like there's a hell of a lot of catalase, or there's just maybe one or two. I don't know. It converts millions per second, so there could be not a ton of it there, or there could be quite a bit. But I can tell you where it primarily resides is in these layers, which are the membranes. So I have a whole new area of investigation.